Dear students, welcome and thank you for tuning in. In this video, we're going to talk about RNA translation into proteins. In the previous video, we talked about DNA transcription into messenger RNA. DNA was transcribed into messenger RNA in order to protect our genetic information. And then the messenger RNA is moved outside the nucleus in order to be translated into proteins. Proteins are made of 20 specific amino acids. Amino acids are what makes proteins. Proteins are functional and structural materials, so they can perform a function like, let's say, insulin can allow sugar to get into the cells for metabolism. Collagen is a structural protein that makes up the skin and muscles and other connective tissue. To summarize what we learned before about proteins, they're made of 20 amino acids. And the amino acids get chained together as a chain. Each protein has a specific sequence of amino acids and number of amino acids, and those amino acids will make and create the functional protein. So amino acids are molecules that contains basic amine group and acidic carboxyl group and the side chain. And those are the 20 amino acids that you can see in the schematic here. We have 20 of them, and they vary in structure in the structure and in the side chain. And the side chain is what determines how the folding of the protein is going to be. And functional proteins have specific structure, specific folding in order to show the active site of the proteins. This system of amino acid is 2 billion years old, so it's conserved for a long time. This is a schematic that shows how the amino acids are linked together to create a protein. They have to be in a specific sequence. If any of those amino acids is out of place, the protein will not be functioning, and that's very important. So DNA is transcribed into messenger RNA, and then now we're going to talk about translating the messenger RNA into protein in order to create the functional protein. And if you look at this schematic, you can see that we have on the top, we have 4 amino acids. So methionine, aspartic acid, leucine, and tyrosine. And you can see that we have the green and the blue color side chains. So what happens is that we are going to study a polymerization reaction where these 4 amino acids should go linked together. And the byproduct of the linkages of the amino acid together to create this chain that's going to make our protein later on is H2O, is water. So proteins are polypeptides that made up of regularly repeated backbone with side chains that impart distinctive physical and or chemical properties because of the charges and how those charges will affect how the protein will be folding on itself and what is the active site that is being exposed and is it functional or not. So this is how proteins are made from amino acids. Now to go back to what we learned before, so we start with our DNA. The DNA is a double helix strand. It's protected in the nucleus. And we said the DNA consists of genes, which are specific sequences of the bases, nucleotides. And then we have spacing, which are non-coding DNA parts. Let's focus on the genes, which are the parts of the DNA that can become, they can have the genetic information to become proteins. So let's say we have this gene in step 2, it's a DNA coding where it starts TAC, and then this tyrosine, adenine, and cytosine. And then you have CGC, which is cytosine, guanine, cytosine. And you can go, each 3 of them is going to be the code for a particular amino acid. So it's packs of 3s, and that's how we translate the messenger RNA into proteins. So each amino acid should go with the code for that amino acid is 3 bases. So we say that we have the DNA strand. So we start at the top of 1, we have double helix, then a signal comes into the nucleus saying we need to upregulate a particular gene to create a specific protein. So the double helix will detach, and we have something one strand instead of 2 strands. So this one strand, we talked about RNA polymerases, which are they going to create messenger RNA, which is the number 3, step number 3 in green. It should be a code from the DNA And you can say that tyrosine is going with adenine, adenine is going with uracil. Remember that A and T goes to adenine and tyrosine go together in the DNA, but in the RNA we don't have tyrosine, we have uracil. And so in the first block you have AUG, which is a start codon, and then and it mimics and it pairs with the top DNA in 2. If you go to the next one, you have GCG in number 3, the next code, and that mimics what you have up at CGC. So cytosine going with guanine, guanine going with cytosine, and cytosine go with guanine. And this is how the messenger RNA is a complement of the DNA strand. Now, this messenger RNA, the green strand, it has a start and stop. And the start and stop is where the gene starts and when it stops. So in order to, let's say, create insulin, this is what we need for insulin. And then it gets chopped. And this messenger RNA has to go out of the nucleus. Once it's out of the nucleus, we have this something called tRNA, which is transfer RNA. And this tRNA is the thing, the shape that have just like the cross. So we know that we need to get amino acids to create the protein. Now, amino acids are floating inside the cells. Amino acids comes from food, from eating, meat, for instance. Those amino acids, they don't float alone. If we want to bring them together and start chaining them to create our protein, they will be attached through an enzymatic reaction to a tRNA, which is step 4. So the tRNA has 2 sides. One side will hold the amino acid, which is the bottom, the circle, and the other side is an anticodon to the messenger RNA. So if you have UAC, and it's step 4, if you have UAC as a messenger RNA, the tRNA, which is the transfer RNA, which the cross side, the cross shape, has 2 sides. One will have the methionine, and the other one will be a complement to UAC, and it knows that that's UAC. The CGC, another tRNA, has another anticodon that's only can attach to CGC, and would have alanine, the ALA, and amino acids. So each code will have a tRNA that has an anticodon that is attached to that particular amino acids. When those amino acids get aligned together, they will detach from the tRNA, and then you go to step 6, which is the peptide, where you have attachment between the sequence of the amino acids that were coded in the messenger RNA. And that's how we create the polypeptide that's going to fold on itself, to create the protein again later on. This is another schematic of tRNA. You can see that at the top you have the ACC, which is the yellow side. Both the left, right, and are tRNA, but just in different schematics, different shapes, different point of view. The amino acids will be bound to the tRNA on the upper side, on the yellow side, and the anticodon, which is the red side, the reddish side, is the one that is going to be attached to the messenger RNA. And each tRNA will have a specific amino acid, only one amino acid. And that amino acid will look for its codon, and the anticodon is going to attach to it, and then we have the sequence of amino acids in the order that we need them to be. There's another schematic. So if you look at the top here, you can see that we have the DNA strand, one strand DNA. On the bottom, the RNA polymerase is it's transcripted into messenger RNA. The messenger RNA is going out, and this is the code for uracil, thymine, adenine, guanine, how it goes. And then we have the ribosomes. Those ribosomes are going to, you're going to have the ribosomes as a protein that's going to link the tRNA to the messenger RNA here. And you can see this here is a one tRNA. This tRNA holds methionine, and this code here is for another tRNA that holds phenylalanine, and then the other one will be a tRNA. It's have anticodon for this codon of the messenger RNA that have leucine, and then each one the ribosomes will allow the tRNA to come attach and then the amino acids will be very close to each other on the top, on the side, the right side, and then the reaction will happen for them to attach to each other to create our polypeptide. And then there will be an enzyme that will deattach them from the tRNA. So DNA, messenger RNA going out. We have the ribosomes. We have also the amino acids attached to tRNAs, the transfer RNA, and tRNA will come to their place exactly where they should be, and then the amino acids will attach to each other, and that's how we form the polypeptide. Another schematic: DNA. This is messenger RNA. Messenger RNA is going out. We have the ribosomes complex. The ribosomal subunits, they will come and they will create this structure. We know that we have tRNA. The tRNA usually attached to a specific amino acid if it has this anticodon, and the ribosomes will bring the anticodon because they're close to each other. The tRNA with the amino acids will come to the anticodon and get close to each other. The previous tRNA is there. They're very close. Now the amino acids are very close. They will attach to each other, and this will deattach from the amino acid, and you have your peptide. You have your peptide sequence. To show it in a schematic, so we have ribosomes, messenger RNA, tRNA, the methionine, and that's the codon and the anticodon attachment. Then another tRNA comes in. They attach the amino acids together. The tRNA get attached. The ribosomes moves to the next three, bases. Again, that's how it's continuously going until we finish the messenger RNA. So once the ribosomes finish the messenger RNA, that's done. Again, keep in mind that the ribosomes and this process should also be initiated by another molecule, another protein that should stimulate and signal the activation of this process. This is our nucleus, and what happened is that the polypeptides, the sequence of amino acids, are made actually in something called the rough endoplasmic reticulum, which is one of the components or organelles in the cell that are very close to the nucleus. Those polypeptides is getting out actually. So after it's been chained together, all the after we have all the amino acids changed together, they will fold on themselves based on the side chains, and then the rough endoplasmic reticulum is going to encapsulate them in a membrane, which is a phospholipid membrane, and then it will go out from the rough endoplasmic reticulum, this bud or this sac that holds all there's a number of those proteins. It will go to the Golgi complex, and it's another process in the Golgi complex where the protein will complete its folding on itself, and then it will be encapsulated in a Golgi vessel that is also just like a membrane, but it's inside the cells. And those Golgi vessels that they have the proteins, if the protein is correct, is the right protein with the right shape and the right number of amino acids, it will be secreted. It will move to the membrane of the cell and it will be secreted outside. Now, if the protein is damaged, and if we have sequencing of those amino acids, if we have the wrong amino acids, those proteins will be flagged and they will go to the lysosomes, where they will be degraded and they will be dissolved, because the lysosomes are organelles that they have low pH, which is acidic, and they will destroy bad proteins or non-functional proteins. So after we create our protein, this is a checkpoint where the Golgi complex checks that the protein is functional, or the one that should be the one that we want. And there's a lot of different mechanisms of how this is happening. We're not going to go into this in this lecture. Everything around the cells are proteins made from the cells. So this is a delicate process. There's a lot of different proteins, a lot of genes, and the feedback control system that control this orchestra is very important. This is another video that tells us how the protein is made, and then it goes into the Golgi complex. where it's checked if it's correct or not, different stages. If it's okay, then it goes to outside and secreted outside of the cells. So this is the dynamics of protein production inside the cells. I hope this was helpful. I know this is a long video, but I wanted to keep my train of thought in place. Thank you for watching. Until the next video.